Alright, so my computer decided to update in the middle of my recording the lecture, so we will start here from cell cycle. And basically, if the cell cycle is controlled or regulated normally with all the checkpoints, if there is something wrong, such as lots of DNA damage, say for instance, then the cell will default to apoptosis. So apoptosis, you saw this diagram in chapter 24, is a regulatory pathway that tells the cell something's wrong. We don't want you to keep reproducing. It's time to die. Okay. So, if cell cycle signals are not correct, if anchorage signals are saying there's nothing to attach to, if density dependent signals are saying the cell don't divide anymore, cells will undergo apoptosis in order to keep that level of new cells, dying cells. And apoptosis is a nice, a neat programmed cell death. And this just kind of shows what's happening. The key here is there's no inflammation. Okay, so your cells are dying all the time. When you were a little embryo, you had webs between your fingers and your toes. And apoptosis was triggered to remove those cells that made up that web so that you had individual digits. So apoptosis is a very normal pathway for maintaining that balance between growth and cell death, and for getting rid of cells, more importantly, for getting rid of cells that are damaged. Many cancer cells have mutations that somehow block this signal for apoptosis. So they might have a mutation in BCL2, which normally inhibits the apoptosis pathway. Okay, that's just one example. So cancer cells can kind of avoid apoptosis due to mutations. A couple other characteristics of cancer cells. Remember that what we're really talking about is DNA mutations and a term called epigenetics, which we're not going to get into, but we get into in genetics, which is changes in control of gene expression. Okay, so when we talk about proto-oncogenes, oncogenes, and tumor suppressors, I'm going to tell you that a lot of them are overexpressed, and that can be due to an epigenetic change or due to a DNA mutation. So a couple other terms that go along with cancer is clonal expansion, clonal evolution. Clonal just means the same. So a clone is identical. And so what this is trying to illustrate to you is a lot of times in cancer, certain cells will divide. And it's kind of a survival of the fittest, right? The cells that are best at dividing are going to take over. But that doesn't mean that all tumor cells are made up of one cell type. And that's what can make it tricky to treat cancer. Because if these cells, say, have a certain protein they express that we can target with chemotherapy, for example, it can wipe out these cells, but it might not wipe out the green or the brown because they might not be expressing the same proteins. And so the important concept here is genetic heterogeneity. Hetero means different. Homo means same, right? You learned that in genetics. So this means that one tumor can be made up of multiple cells with different genetics. And that's going to mean they have different phenotypes. They're expressing different proteins and molecules, and that can make cancer very tricky to treat or get rid of. So, another reason cancers are so successful. So just to summarize, some characteristics of cancer. The main thing is that they continue to divide, right? They're constantly dividing, and this can be because of many reasons. Because telomerase is on, okay? Because they're not responding to cell signaling, so cues from the environment, such as density and anchorage. Cues. Environment. Cell cycle control is lost, which means most of the time the checkpoints aren't working, and this is again where we're going to talk a lot about tumor suppressors and proto-oncogenes, or there's some kind of block in the apoptosis pathway, so the cells don't die. All right. So, what makes cancer so deadly? The main thing, or one of the primary things, is that it can continue to divide or grow. Right? This is tumors. Okay? And we talked about, or I've mentioned, that this can put stress on tissues and organs. They're able to grow because of angiogenesis, and that's what we're going to talk about next. So, cancer cells have a means of getting their own blood supply. The other reason cancer is so deadly is because of metastasis, or I should say ability to metastasize. Metastasize? It's always a word too. Alright, which means the cancer can move to new locations in the body. Right? Kind of like chasing those little bunnies that are outside all the time. Right? They just keep moving and moving and moving. Makes it so hard to control. So let's talk a little bit about angiogenesis. Okay. And the key to angiogenesis is that it's tumors promoting growth of their own blood supply. Okay, and why is this important? Well, you see this mass of cells, they need to be able to get oxygen and nutrients and get rid of waste. That's the function of the blood. So without blood vessels, the tumors can't grow. So angiogenesis happens in tumors greater than 1 to 2 millimeters. And I find this just fascinating, because if you pull out a ruler, 1 to 2 millimeters is pretty dang small. And I couldn't understand why they were so worried when my mom had a 5 millimeter tumor in her breast. I was like, 5 millimeters, who cares? That's not going to do anything. Well, what that indicated to them was that the tumor had its own blood supply. and we're going to talk about in a second, that having its own blood supply means tumor cells have access to metastasize. So, again, not all tumors, benign ones, don't metastasize, but there's that risk when you get these large, 1 to 2 millimeters, large tumors. And I did a little calculation. 1 millimeter can be like 70 to 80 cells. So if you start thinking about lining up 75 to 80 cells, that's a lot, right? And if they all need blood and oxygen, or they need the blood because of oxygen and nutrients, then it makes sense that they need to get this blood supply. So how do tumors do this? Well, the tumors secrete growth factors like VEGF and FGF. So VEGF is vascular endothelial growth factor. FGF is fibroblast growth factor. And the tumors are secreting these, and then these cells are endothelial. So epithelial is our skin, endothelial is the lining of our blood vessels. Okay. So what's happening is that the tumors are secreting this growth factor that's binding to the receptor on the endothelial cells and telling the endothelial cells to proliferate, to divide, to migrate, to move, to increase permeability. We'll talk about that in a second. And results in angiogenesis, new blood supplies being formed. So I like this signaling pathway. Again, more information than you need, but some of the things that happen once these endothelial cells are stimulated, excuse me, by growth factors, is not only do they start to divide, they migrate, which means they move. And one of the ways they do this is they secrete MMPs. And hopefully you remember these, matrix metalloproteases, which are important, which degrade the extracellular matrix. So these cells are not only proliferating, so they're dividing, but they're able to move because they secrete these proteins that start destroying the extracellular matrix, and it allows those cells excuse me, to move to that tumor. Oh, I didn't do this right. Okay, hold on. Right. But I'm going to erase some of these because it didn't quite work the way I wanted. There are natural angiogenesis inhibitors. Okay, so your body is naturally producing things that monitor or prevent blood supply. So you don't want your blood vessels going everywhere. So there has to be this balance between where you need the blood supply to go and stopping these signaling. So angiostatin, endostatin, thrombospondin are all natural angiogenesis inhibitors found in the extracellular matrix, and there's this kind of ratio that plays between what the tumor is producing and the angiogenesis inhibitors as to whether these blood vessels will be made. And angiogenesis inhibitors in general are a class of chemotherapy that they give patients to try to block that blood supply to the tumor to stop it from growing. Okay. So cancer is an issue because you have this uncontrolled cell growth, it can promote its own blood supply, and by having this blood supply, now you can get cells breaking off from the tumor and moving to different areas in the body, and that's called metastasis. And this can be through the blood or through the lymph. So lots of times, so my mom with her breast cancer, they did radiation to kill the tumor, but they also removed some of her lymph nodes that were right around that tissue because they were afraid cells may have broken off from the tumor and moved and implanted in the lymph nodes, so metastasized. So, excuse me, metastasis is simply cells breaking off, moving through the blood or the lymph to a new place, exiting, and now growing. So you have a tumor, secondary tumor on a different location in the body. Your book talks about some interesting, like, where these normally happen, in the lungs, in the liver, depending on where the what type of cancer it is. So it's kind of interesting to read that part of your chapter. The term invasion just means when the cancer cells are moving into local tissue. Okay, so invasion is different than metastasis. Metastasis means the cancer is now located in a totally new, distinct spot in the body. Okay, so you can have invasion where you're damaging the tissue around the primary tumor, or you can have metastasis where a new tumor has formed excuse me, in a new location. And your book also talks about that a lot of these cells don't survive in the blood vessels, in the bloodstream, and they're not able to colonize and proliferate in a new spot. And that's kind of where this idea of tumor heterogeneity is important, because you might need to try out some of these different cell types to see which ones can metastasize, move, and make a tumor somewhere else. So what's happening is first you're going to have some invasion where you're going to kind of move from that tumor into local tissue. And what's happening here is you have a loss of cell-cell adhesion proteins. Such as E-cadherin. Remember, this is part of adherens junctions, right? And they hold tissue together. So if you start downregulating, you lose these proteins, cells can break off from that tissue. You also have something called chemotaxis, which means the cells are attracted to some kind of signaling molecule, and that's what attracts them to move away from that primary tissue. So you get cell mobility, which means they move, and that has to do with chemotaxis, and it has to do with activation of the Rho family GTPases. So if you remember from our cytoskeleton chapter 13, the Rho family GTPases stimulate actin remodeling. So if you've got actin remodeling, you can get these cells to move. They can make filopodia, they can make lamellipodia, and they can move to different sites. And the other thing that's happening with these cancer cells, just like in angiogenesis, is that the cells produce proteases that break down the basal lamina This is like for skin cancers, endothelial, and the extracellular matrix, right? So, for instance, plasminogen. Oops. Activator. Right? We'll start activating these proteases that will start breaking down this matrix so that the cancer cells can move. They'll activate matrix metalloproteases. Right? So the cancer cells are able to turn on genes, express proteins, or decrease protein expression that allows them to break free, to invade, and then metastasize and colonize or kind of settle down in another location of the body. All right, I'm going to stop there, and then we're going to come back to 26.3, what causes cancer.